There is no evidence, like zero evidence, of any causal link between the microbiome and autism. We've been through this replication crisis where we've learned if you do studies like this with small samples, with loads of degrees of freedom in how you analyze the data, and with no correction for proper stats and no replication, you're just going to flood the literature with false positives. And that absolutely happened in genetics and, neuroimaging and social psychology, for example.
What are the incentives here for the scientists to be making what seem like very obvious methodological issues? It kind of shocked me how poor the study design was.
Yeah, and it shocked me as well.
Dr. Kevin Mitchell is a neurogeneticist at Trinity College Dublin whose work has fundamentally reshaped how we understand the brain, how it wires itself, why it varies so much from person to person, and what that means for individuality, behaviour and consciousness. He's the author of Innate, a landmark book explaining how the architecture of the brain makes us each feel unique, and Free Agents, his bold argument that evolution built minds with real biological free will. Free Agents is included in my free ebook 26 Neuroscience Books for 2026 that you can download below. Kevin has also become one of the sharpest voices in modern neuroscience, calling out hype, demanding rigor and dismantling shaky claims in the social sciences, genetics and, as we will see in this episode, autism and the microbiome. This episode is not meant to be anti-science, but it does expose a glaring problem with academia, how entire fields that seem at first glance to be strong can emerge from really poor and shaky foundations, and how harmful that misinformation can be for the people genuinely suffering from these conditions. Subscribe before another underpowered mouse study convinces your gut that it's running the show.
You'll see these reports come out of some factor that's linked to autism, schizophrenia, depression, cancer, whatever, right? And then over time, other people might report something similar, and then some studies come out that don't find that association, and then finally somebody does the study properly. They control properly for all these possible confounds. Then, in this case, the Tylenol thing just disappears. It's just not there at all. There's no association. The sample sizes for some of these other papers, again, they were tiny and way, way lower. The recommended samples that have been put out by people in the field studying these things, who develop these behavioral tests, who say, look, you need multiple replications, 20 animals per group, per sex, per age, not 5 animals, right?
Five animals. That's the one that I read. It's like I should at least have more fingers than there are subjects in your study.
Yeah, that's the frustrating
I was like, how
bit of it, right?
But that's where it was kind of like, this seems almost too strange not to have some malicious incentives somewhere. Because I'm like, that seems too crazy.
The overall conclusion from them is that when the experiments are done properly, you don't find any effect.
Dr. Kevin Mitchell, a huge welcome back. You were number one on the giant shoulder, and I just couldn't be more appreciative for you coming back, and I'm so excited to have you.
Sure, it's my pleasure, Evan, and great to see the podcast, going from strength to strength. It's brilliant.
Well, I have so much to thank you for it. Probably the first 10 episodes we did were from introductions that you made. So you were not only number one, but in extended, you were also maybe 1 through 12 in just the introductions that you allowed. And then it's really the momentum from those that has allowed me to, speak to Michael Levin, Anil Seth, David Nutt, Peter Godfrey Smith. Like, the list kind of breaks my mind thinking about the incredible scientists. So I've so much gratitude to you for
brilliant.
that.
I know, it's my pleasure to do that. And, especially these days, the more good, solid, sensible science communication we get, the better. So I'm delighted.
Yes, thank you. So you're coming out with a new paper. The title is Conceptual and Methodological Flaws Undermine Claims of a Link Between the Gut Microbiome and Autism. Super interesting topic at the moment. I feel like every day you almost see news reports of like the graph of increased autism prevalence, and people kind of just throwing every explanation under the sun at what might be increasing that graph. I saw a pretty funny one of RFK, time that RFK has been on the planet, and autism. And it's like, the only thing to conclude from that graph is that RFK himself causes autism. Well, from that graph, I mean, there's no arguing it. So, contentious debate at the moment. What really compelled you to write this paper?
Yeah, well, a few things. I mean, like you say, it's very timely and topical, right? It's on people's mind. It's an important topic. What causes autism, right? We want to know. Lots of people working on it. This is a condition that affects, millions and millions of people worldwide and their families, and can be really devastating in its most severe manifestations. So we want to know, obviously, as scientists, what's causing it. But also, there's this background of misinformation, I think, out there where there's all these kinds of people, leaping at these different potential causes of autism. And, vaccines are the most infamous example there, where the damage that was done from those initial, terrible, fraudulent studies actually is just huge. It's incalculable, and we're still reaping the impact of that. You can see it in the policy in the United States at the moment. So, it just is, to my mind, really important to be doing good, rigorous science here. All the more important because there's loads of pseudoscience out there, and you can't fight pseudoscience with dodgy science. It has to be good. And so, it just is really important, I think, that we collectively apply standards of rigor and reproducibility and, a kind of normal degree of scientific skepticism to these kinds of claims, so that the body of literature around causes of autism is as solid as we can get it.
Right. Like, I couldn't agree more, of course. We even had the Tylenol news story, was that 3 or 4 weeks ago now? Like, what are your thoughts on that? Like, that went everywhere.
Yeah, that's just an example of terrible, terrible work that gets taken out of context and then massively extrapolated. It's a good illustration of some of the problems. So these are, the first reports about that came from very small scale epidemiological studies. And when you do an epidemiological study, you're basically looking for some factor that's more prevalent in people with a condition than without. So if you look at, people with lung cancer versus without, then smoking is an exposure that's way, way more common in the people with lung cancer. So that's how we do epidemiology, right? Now, the challenge is, first of all, to find things like that, that have a really big effect that you can be quite confident in, that it's a real statistical association. And then secondly, that it's a causal association, because we don't know that from the get-go, right? It could be something else is causing lung cancer. So maybe like heavy drinking might have been the cause of lung cancer, and it could have just been the case that people who are heavy drinkers also tend to be smokers more often than others, right? So there's all these potential for confounders in these epidemiological studies, which we'll see is absolutely the case with microbiome stuff as well. But what often happens in the literature is that you'll see these reports come out of some factor that's linked to autism, schizophrenia, depression, cancer, whatever, right? And then over time, other people might report something similar, and then some studies come out that don't find that association, and then finally somebody does the study properly, right? So they go to Denmark or Sweden or somewhere like that, where they have a national register. They got everybody's data. They can look at huge cohorts of millions of people, not dozens or hundreds of people. And then when they look at that and they control properly for all these possible confounds, then in this case, the Tylenol thing just disappears. It's just not there at all. There's no association. And, it's an absurd hypothesis on its face anyway, given the timing of the introduction of Tylenol, that it just doesn't match the epidemiology and so on. But the other factor there that's also really important is that even the initial studies showed a tiny effect size. So what that means is you can work out, okay, how much more frequent is Tylenol usage in people with autism. In this case, it was mothers using Tylenol and their offspring having autism, right? Versus ones without autism. And you compare the size of that frequency difference, and that gives you an indication of the the increased statistical risk that's associated with the exposure. And that's just a statistical thing. It doesn't actually say anything causal, but that's the basis for a causal hypothesis, right? And when you do that for
Yes.
the Tylenol thing, like the risk is, I don't know, it's 1 point something, right? It's not 50 times greater frequency. Like there's no way that it could explain what it's purported to explain here. So
Yes.
it's an indicator of where you've got this study, these studies come out, you see a headline. The headline is simply Tylenol is associated with increased risk of autism, right? And most people just don't.
I don't even think it's that many words most of the time. It's just Tylenol causes autism. Like what you just said there was almost twice the nuance of the average headline.
It's true, actually, right? Because they go beyond the association to assume causality. They assume that it's an important cause if it were a cause, right? So they ignore the fact that the effect size is actually tiny. And to begin with, they just take at face value these statistical associations from small studies and ignore the huge, properly done study with millions and millions of people that says, no, there's nothing going on here.
Yes. And I think whenever a retraction is made, if a retraction is made, it gets like 1/100th of the publicity of the original headline and news story that went around with those initial very low power. Like I was shocked at the size of the samples in some of these experiments that I was reading through. It kind of was blowing my mind.
Yeah.
And I could totally see how that was generating all these headlines. And then these larger powered studies with better methodology were done and often didn't find any effect. And of course, that's not going to get the headlines. That's not going to go viral on X. That's not going to have YouTube communicators talking about.
No findings or negative findings, a lack of replication of something is not a huge big story. No one in the newspaper is going to write that, right? They love a simple story where there's this this really complex disease or condition, right? I mean, let me just clarify actually that, we think of autism as a disorder. And the reason that it's called that is because people suffer from it, right? So, when they seek medical attention for it or some other kind of intervention for it, that's basically just the pragmatic reason why it's called a disorder, right? So it doesn't have to be any kind of more judgmental tenor to that word. It's just a very pragmatic sort of a description. And of course, it has a huge range. It goes all the way from people who will, never speak, really sort of serious behavioral issues will need 24-hour care potentially for the rest of their lives, to people who are you might say are just a bit odd, but who get on fine. They function perfectly well, right? So so it's important just to say that. But when you have a condition like that, of course, people are looking for causes. And a good news story for a journalist is, I can say a study was done and it found this cause. Game over. That's it, right? That's just a neat story. You don't need tons of background. You don't have to understand molecular genetics or neural development or anything like that, right? You can just say, oh, this, whatever it is, chemtrails or fluoride in the water, whatever it is the cause, and that's it. And so in terms of the science communication, part of the problem is that there's this apparent void, right? It looks like we don't know what's going on, and into that void you can throw all kinds of hypotheses, right? And it turns out that we have a really good handle on what's causing autism. It's really, really highly heritable condition. It's super genetic in that sense, but it's genetic in a really complicated kind of a complex way. So it's not like it's one gene always, like something like cystic fibrosis, where like it's always the same thing. It's not like that. It's more like an umbrella term like intellectual disability, where intellectual disability as a diagnosis can be caused by mutations in hundreds and hundreds of different genes. So it's really heterogeneous, genetically speaking, and complex. There's interactions between genetic variants and so on that, give kind of different levels of risk and severity of the condition. So that's a hard story to tell, right? That doesn't
Yes.
make a nice, tidy 3-column newspaper story, right?
Right.
That requires well, hours and hours to really even describe what's going on, as well as, you know, years of study really to actually understand it in depth. So that's a
Yes.
challenge, right, for scientists, is to try and describe what's really going on when there's this sort of other kinds of work that's going on that purports to give a nice, simple, clean answer that also is, in many cases, a point of potential manipulation, right? That's the other thing that
Yes.
appeals for a lot of these things, is that with genetics is like, well, there's nothing you can do about that now. But if it's something like the microbiome, well, then the promise is, okay, well, maybe we can treat that. And so that becomes an even more attractive kind of story, psychologically speaking, not just because it's
Yes.
simple, but because it's hopeful in that sense.
Yes. Let's go into those. So I've been trying to drink less alcohol. And honestly, it's had a pretty big impact on my health. I'm sleeping better, I'm exercising more, and my business is growing. The data suggests that people are drinking less alcohol than ever, which overall I think is great, but I'm still looking for a little treat at the end of the workday or a hard workout. And that's where Sentia comes in. Sentia is a 0% alcohol drink that feels like you're drinking something special without any of the downsides. It's created by neuroscientists and it's designed to give you that relaxing buzz, the same kind of feeling you get from alcohol, but without the hangover, brain fog, or impact on sleep quality. And it tastes absolutely delicious. They already have a GABA red berry, which is perfect to mix with a tonic or orange, or GABA black, which is awesome with Coke. Personally, I've been really enjoying the GABA gold with Fanta lemon on ice. That's actually genuinely delicious. Like, I'm not even play acting now, it's actually just really, really good. So you might be wondering, how does a drink with 0% alcohol give you that buzz? It almost sounds too good to be true. Well, this is the really cool neuroscience part. The secret is GABA, the brain's natural calming agent. And the different GABA flavors have different cognitive effects. Sentia's founder, Dr. David Nutt, has published over 500 independent research papers and written 34 books on neuropharmacology. What Dr. Nutt realized was that we can harness the same calming, relaxing effects from alcohol without any of the side effects. I genuinely think this could be the future of drinking. And they're also just releasing their first beer and stout, which as an Irishman I am dying to compare to Guinness. Sentia are offering my viewers 10% off their first purchase, and I genuinely could not recommend it more. I think even if you're looking to just substitute your one gin and tonic on a Tuesday with Sentia, the cumulative health benefits over time will actually be very noticeable. So check out Sentia, again, 10% off at checkout. What are the most common lines of evidence that people usually draw, scientists in the field draw, between the gut microbiome and autism? And maybe steelman the position a little bit first. We'll set the foundation of like what the literature really says, what are the strongest pieces of evidence, and then we can kind of go into the content of the paper.
So the background to this is this idea that things that are going on in our gut, the bacterial populations that are producing various metabolites and so on, can in some way inform what's happening in our brains and affect our moods and affect our psychology and so on. And if you've ever had an upset stomach, you realize, that can affect your mood and so on. That's a perfectly sort of everyday kind of occurrence, right? The broader hypothesis is that there's something systematic that's going on, where there's differences in people's microbiome over, that are stable over time, that explain something about their psychology, even to the point of potentially being causally implicated in things like autism. And now, autism is just one of the things that's been where these associations have been proposed. And it's every psychiatric condition you can think of: schizophrenia, OCD, PTSD, depression, anxiety, but also things like, eczema, asthma, obesity, heart disease. The list just sort of goes on and on, where something about this gut-brain axis is implicated in these conditions. So it's actually hard for me to really steelman the hypothesis, and I'm going to
You put so much work into discrediting the field in this paper, like
not just because of that, but because I find it kind of it's an extraordinary suggestion, right? It's an extraordinary hypothesis to begin with. I think that's part of its appeal, is it's so sort of out there and novel, and it sounds like it's kind of overturning these paradigms that everyone's been working in. It's like, okay, what if I told you that rather than your brain affecting your mind and explaining in some way your psychology and even psychiatric conditions, what if it's actually what's going on in your gut? And it's like, it's like, okay, that's sort of a cool idea, right? It sounds new and kind of sexy. And it's like, look, you didn't know there's so many bacteria in there. There's more bacteria than there are cells in your, human cells in your body. And so you can kind of sell it that way. But, in terms of actual evidence, it's really light on the ground. So
yes.
you can say there's a hypothesis there that's worth considering, and that's fine. And at some point you should say, okay, well, having done all this considering, what does the evidence look like, and now how does that hypothesis stand, which is what we were trying to do.
Yeah. I think one of the things that I heard that if you look at it in a vacuum without context seems interesting, that there is consistent differences in the gut microbiome of people with autism compared to controls. That is true in a vacuum, right?
I mean, not even that, actually, to be honest, because, well, that's partly what we found, is that any differences that are observed are not consistent. But actually, let me back up, because I didn't do justice to the origins of this hypothesis. So really, it didn't come out of nowhere, this potential link with autism, because one of the consistent findings in autism is that patients often have gut GI problems of some kind or another, right? So at a higher frequency than the general population, and the numbers vary. It's really hard to actually get numbers on that, but one of the studies showed it was like around 33% in autistic patients versus about 15% in control children, and so that was twice the frequency. So looking in that, there's something linking in the phenomenon that we're observing. There's something linking something going on in the gut with something going on in the brain, and there's a number of ways to think about that. One is that something's going wrong in the gut and it's affecting the brain, right? So whatever is happening in the gut is primary, and it's secondarily causing the symptoms of autism, right? That's the sort of general way that this hypothesis is pitched. There's another hypothesis, which is that these are there's a reverse causation, which is to say the symptoms of autism are in some way causing the GI symptoms. And that could be because, say, people with autism are really picky eaters, potentially, right? So something about their behavior affects their diet, which affects their gut and potentially their gut microbiome. So that's a reverse causation scenario. Yes. But then there's another scenario, which is actually much more common for neurodevelopmental disorders in general, which is that genetics is affecting both the gut and the brain, right? And that's why we have, there's so many genetic syndromes. The reason they're called syndromes is because whatever the genetic, mutation is or lesion, it can have lots of effects on lots of different organs, and they're independent from each other, right? And in fact, the gut effects could be effects on the gut nervous system, right? There could be the enteric nervous system could be affected just like the central nervous system is, and so on. So, and we know, for example, that gut, problems are not specific to autism. They're much more common in all neurodevelopmental disorders, even things like Down syndrome, for example. Patients with Down syndrome have a higher frequency of gut problems as well. So,
I didn't know that.
So obviously the arrow of causation is pretty clear there, right? In that we know what the genetic, cause is in that case, and it's clearly affecting both the gut and the brain probably independently.
Yes. Let's go into the first line of evidence then. In the paper, you start off with human observational
Yeah.
data. Talk about the field's attempt to link the microbiome and autism with human observational data. First off, what does
Yeah.
that mean, human observational
Yeah.
data?
So the hypothesis as it stands really rests on three kinds of evidence. There's observational studies in humans, uh, experimental work in mice, and then clinical trials in humans. And we look at each of those in turn in this paper. And so an observational study is like what I was talking about before, just an epidemiological study where you look for, you take people who have a condition and people who don't, and you look for some factors that are more frequent or, in the people with it, or that are different between those two, between the two sets of people, right? So the first of these studies came out about 15 years ago, I think in 2010, where people suddenly there were tools available where they could look at the microbiome in the gut, from the gut of people, right? So basically, you can take a stool sample from people. You can use molecular tools to figure out what bacteria are in there. And there's a variety of different sort of platforms and methods you can use to do that. There's a whole load of sort of technical caveats around exactly how it's done. But the point is, I could take your microbiome and my microbiome, and we could look for any differences between them. And what that data looks like is basically you're doing loads of DNA sequencing of the DNA of microorganisms that are in the gut, and the number of reads that you get that correspond to one species or another is taken as an indicator of the relative proportion of the microbiome that's made up of one kind of bacterium versus another. Okay? And you can group those data in different ways. You can look across major phyla of bacteria, so big groups that have lots of species between them. You can go down to the genus level. You can go down all the way to the species level and look for individual species differences in abundance between people with autism and without. So that's the nature of the experiment. And when you do that, well, first of all, it's probably already apparent one of the problems here is that you get huge amounts of data, right? There's all these different species that come up there, right? Hundreds and hundreds and hundreds. And so you have so many data points that you can compare between your two groups. Now, if your groups are small, then and you're comparing, loads and loads of variables, then you're going to get lots of noise. And in fact, when you do this, just from one person at multiple times, the data are super noisy. Like your microbiome is what's present in your microbiome is fluctuating all the time, depending on what you just ate, depending on what time of day it is, from one week to the next. It's just not a very stable sort of thing in its details for any individual person, right? So you're taking a very noisy measure, very multivariate, and then you're doing loads and loads of comparisons between people. And of course, like I just said, you could do comparisons at the level of the phylum, there's 7 or 8 major phyla of bacteria, and maybe you don't find anything, but then you say, okay, well, now I'm going to look at the level of the genus or the level of species and so on. So there's a lot of degrees of freedom in the way that you can do this analysis, and it's very unusual. In fact, none of the studies that we looked at had a registered plan that they set out beforehand to say, here's what we're going to look at ahead of time. We're going to look at these phyla and these genus and these species, and that's it. They just sort of you can just mine these data, right? So when people started doing those experiments, some positive associations were reported where people were saying, look, here's the microbiome of people with autism, and it's different from that of people without autism, and here's the particular bacterial species or whatever it is that's different, and they could be increased in people with autism or decreased. And that's another example where it's sort of like anything will do, right? It's an exploratory analysis.
Yeah.
So any difference is taken as being important. Now, there's a number of issues with that in terms of the way that these data are treated. So if you're doing loads and loads of comparisons, then you need to correct your statistics. for all the tests that you did. Because if you're just going at our normal, what's called a p less than 0.05 cutoff, that means you're saying there's only a 1 in 20 chance of having seen these data if there was no real difference underlying it, basically, right? But if you do 20 tests and that's your cutoff, well, you should expect at least one of them to hit that cutoff, because that's what it means, right? It's a 1 in 20 chance just by randomness, right?
Yes.
So if you're doing hundreds of tests or thousands of them, then you're almost guaranteed to find something that's most likely just a blip, just a spurious false positive, right?
Yeah.
Now, a way to figure out if it's a false positive, of course, is to replicate it, right? To say, okay, hey, here's my exploratory sample. I'm going to do this. I found these things. And then I'm going to look in another sample and I'm going to test these specific things that I think I saw here and see if they hold up in this other sample. So that's a good way to do a test. And it's not the way that these studies were done, right? They were one-off, no replication sample. Many of them didn't correct for multiple tests. And they had really, really small numbers of people. Some of them had like less than 10 people per group. We looked at we take to be a kind of a seminal set of papers, about 12 papers, maybe a few more, that people cite as the evidence base that there's some differences between people with autism and people without. And the numbers are, they went up to 50 per group, but that was as high as they went. So that's the nature of the evidence that came out. And what you would like to see in order to say, okay, something real is here, is that different people, these different studies would converge on something, right? They would consistently find, if you did a study and you found Lactobacillus reuteri was higher in people with autism, and then I did a study and I didn't find that, or I found it was lower, well, then, you don't have a lot of confidence in that, right? And it turns out nothing consistent emerges. The data are hugely inconsistent across this set of studies that people have done. And yet the message somehow is still, okay, well, you found something. And, I found something. And okay, yeah, they were different, but there must be something there, right? There must be something going on if people keep finding some difference. But, you could look at multiple of these studies and say, actually, they're just contradicting each other. This is just the only consistency is a failure to replicate the findings from any particular study across, all the other studies.
That was really, really wild to me, and just seemed like a huge categorical error for scientists to be making, where papers were finding a difference, but it wasn't the same difference. But they were taking the fact that they found any difference at all as a replication, when clearly a different difference is not a replication. That's just a different result.
Yeah.
But again, that seems so obvious that it almost like it feels weird to even like say out loud. Like, why do you think? And throughout the paper, I was thinking this, like, what are the incentives here for the scientists to be making what seem like very obvious methodological issues, like very basic, like not correcting for statistical tests? Like, these are stuff you learn in college. Like, why? And also, why are they not caught by peer review? Is peer review not supposed to kind of point that out, that the stats aren't done right and there's methodological issues? It kind of shocked me how poor the study design was.
Yeah, and it shocked me as well, especially because we've been through this. I mean, so my work is in neuroscience, genetics, and psychology. And in all of those areas, we've been through this replication crisis where we've learned if you do studies like this with small samples, with loads of degrees of freedom in how you analyze the data, and with no correction for proper stats and no replication, you're just going to flood the literature with false positives. And that absolutely happened in genetics and neuroimaging and social psychology, for example. And so what we learned in those fields is you need to do the studies bigger, you need to do your stats properly, you need to replicate. And it's not easy though, because what had to happen in genetics was that people completely had to change the way that they worked. Rather than me collecting, painstakingly going to the local hospital and recruiting loads of patients with schizophrenia or autism, whatever, to a study where I might end up with a few dozen patients, maybe 100, and I'll do my local study just myself, it was clear that that wasn't working. It doesn't work for looking for neuroimaging differences. It doesn't work looking for genetic differences. And what people had to do was make these huge consortia, which required just a massive sociological change in the field, frankly. In order to do that, you now see these, genome-wide association studies and sequencing studies that have hundreds of authors on it. So instead of me getting my last author or senior author paper from my own lab with 3 or 4 people in it, I could be middle author out of 200, and that just looks really different, right? But that's what had to be done to get rigorous, reproducible results. So part of the degree of exasperation personally, that I was experiencing in reading these papers was like, we know not to do this now from other fields. And it was sort of painful to me that the people in that field hadn't sort of gotten that memo. And I think, partly it's because you get this new technology, and then it generates huge amounts of data, and then people don't quite know what to do with it. So there's, you can expect some teething pains and so on, right? But up to after a point, you have to say, okay, that you're not getting the benefit of the doubt here anymore. There's a right way to do this, and there's a wrong way. And part of the problem, I think, is that within a field, if you have some established papers out there and they did it this way, then someone else comes along and does it that way. It's like, well, that's the way everyone says that's fine. So you get a kind of a culture, and I think that's how these things get by peer review, is because there hasn't been this this culture change which we saw in other fields.
Right. Makes sense. Do you think that GWAS is sort of inherently a flawed methodology? It seems like false positives are a feature of that design. Can they be done correctly? Is there utility in doing this kind of gene association study where you're just comparing thousands of variables? Or do you think this is just a methodology that we really shouldn't be employing?
Are you talking specifically, Evan, about the genetic kind of things like GWAS, or association studies in general where you have thousands of variables?
Let's do GWAS specifically, where you're just looking at the genome and then looking for any associations.
Okay, so what had happened in the field was that before GWAS came along, and GWAS basically allows you to look for genetic variants across the whole genome, where there's sites in the genome that might be, in some person they're a G, in another person they're an A in the DNA sequence. And the G might be at like 70% of people have a G and 30% have an A, something like that. And there's millions of these sites across the whole genome. And those genetic differences can contribute to traits that we have, like differences in height and, other things, but also to susceptibility to disease. So people have looked to try and basically do the epidemiology like what I was talking about. So take people with, say, autism and without, and then look across all of these sites and say, are any of these sites where one of the variants is more frequent in people with the condition than without, right? And then you say that's a risk factor. Now, there's 2 things to say about that make it different from the other epidemiology. First of all, we know that there are genetic risk variants, just because these conditions, we know that they're really highly heritable from twin studies and family studies and population studies. And what that means is that for autism, 80 to 90% of the variants across the population and who gets this condition is attributable to genetic differences, right? So that's our baseline. Right. That's a sort of what I take to be a really solid baseline. And then the question is, can we find the variants that are responsible for that? Okay, but we know there's something there to be found in the first place. The other thing about genetic epidemiology is that the arrow of causation can only go one way. It can't be the case that having autism somehow goes back in time and affects which genetic variants you inherited from your parents, right? That arrow of causation is off the board, whereas it's very much on the board when it comes to things like the microbiome. You don't know which way the association is going, causally speaking, if you find one. So the trick with GWAS is you're doing maybe 500,000 tests, maybe 1,000,000 tests, something like that. And then you have to set your p-value for statistical significance really, 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 really stringently. And that's what people do.
Yes.
And the only way that you can possibly do that is with thousands and thousands, actually hundreds of thousands of people, right? That's what we found over the years, is you can't do this with hundreds of people. You can't do it with actually even thousands for most cases. It literally needs tens of thousands or hundreds of thousands of people to statistically significantly detect these kinds of effects, right? And
Yes.
so what people have done is make these massive consortia, loads and loads of subjects into their GWAS, do the stats properly, do the replications, and they have found replicated associations, and they implicate genes involved in neural development, which makes sense. They implicate genes involved in neuroplasticity, which makes sense, and they hit some of the same genes that we find where really rare mutations can cause a condition that manifests as autism or other kind of effects. So the GWAS is really solid in my view.
Okay.
But it has so far only explained a percentage of the variance that we know is attributable to genetics. So it's still very much a work in progress. And when people look at it and say, but look, you're missing some of the heritability, therefore there must be some environmental reasons for that, that's just a non sequitur. That just doesn't follow. Right?
Yes.
Now the contrast with the microbiome stuff is pretty stark, because there you're also looking at at least hundreds or thousands of variables, and the all of the sins and flaws that were there in early genetic studies are being repeated in the microbiome studies. And there were some really interesting papers that came out in the last year or two, which showed that in order to find robustly, reproducibly find real signals where we know there's a real association with some factor and the microbiome, things like body mass index, age, diet, smoking, diabetes, these are all conditions that we know affect the microbiome. In order to find those associations, it required samples with thousands of subjects,
Yes.
right? Not tens of subjects, which is the level that most of the early microbiome papers were using. That matches with things that we know from neuroimaging. The paper came out recently, again, showing the same thing. You can't just scan a brain and look at thousands and thousands of parameters in 20 people or 50 people. You need 2,000 people or 5,000 people to do this do this right. And so that's where we are.
Should there just be sample size guidelines for different methodologies that are sort of established, well researched? Like, as you said, a study came out showing that we need these sample sizes so that if a paper is going for peer review and the sample size just doesn't match the guideline, it's just not published, because it's going to cause false positives, bad headlines, bad science, contribute to misinformation in general, if we know that there is good sample sizes for different methodologies.
Yeah, absolutely. And that's what happened in the genetics field. It's what's happening in the neuroimaging field. And actually the best place to catch it is at the prior to doing the research. If someone's proposing to do the research, they have a grant application in or something like that, or even they're just thinking about it, that's where this kind of critique should come in terms of peer review, to say, look, your study is massively underpowered. You're not going to find anything. Anything you do find is likely to be noise and worse than doing nothing. Right. And, it's, I mean, I've had this discussion with people who are doing neuroimaging, and they'll say, but, I can't, I'm a small lab. I can't possibly collect thousands and thousands of people and scan them all. I can only do dozens of people. So, what am I supposed to do? How am I supposed to train my graduate students if I can't do my small bespoke artisanal neuroimaging study? And, I'm sympathetic to their plight, but I absolutely don't buy that as an argument. It's just a waste of time.
Do a different study.
Yeah, just do something else. It's just a waste of time. It's a waste of money. It's a bad training to do that.
I think nothing highlighted this sample size issue more than the next line of evidence, which blew my mind. Some of these sample sizes I could count on my two hands. I was like shocked. So do you have anything else that you wanted to cover there on human observation, or we go to mouse? We go to mouse model.
Yeah. So let me say a couple other things about the human stuff, because there's a few things that are important. One is that so when people did these various studies, in any given population sample that they looked at, there were some differences that they reported, right? Question is like what to make of those. And there's a few options. One is to say, these are statistical blips. That's not signal. I should just not pay any attention to it, right? That's actually where I would just stop. That's where I would start and stop and just say, actually, you don't need to explain this, right? But let's take them at face value and see, well, what should we make of those? One possibility is that there's a real causal association. But another is that there's a reverse causal association, right? That actually some confounders are they're driving the microbiome differences, whatever they are, that you happen to see in your population, right? And so there's good evidence from a bunch of other studies that changes in diet, especially in people with autism, who tend to have quite restrictive diets, can lead to some changes in the microbiome, right? So if there is a change that you see, then it's important to just not conclude that it's causal in the microbiome to autism direction. It could be in the other direction. And another reason to think that a lot of those positive findings might be due to confounding is that when people did sibling controls as opposed to unrelated people, a lot of those supposed differences just went away, right? The people with autism were not different from their non-autistic siblings. And so, I think that's really strong evidence that whatever this sort of sea of reported findings was actually probably mainly driven by confounding and, isn't a robust finding. So I think those are important. And then the last thing I'll say, which is also really crucial and often overlooked, is to think about when there's an association, what's the effect size, right? How big of an association, how strong of an association is it? Because, what we know, say, for lung cancer and smoking, that's a massive statistical effect. When we do twin studies and so on, when we say, like, your risk of having autism, a person's risk of having autism is about 80-fold higher if they have an identical twin with autism. Right? This is a really, really clear indication of a genetic effect, that carrying that same genome is a risk factor, and it's a massive, massive risk factor. Whereas, if you look at the genome-wide association studies that are looking at all these common variants across the population, individually they have tiny effects, like on the order of 1.1 fold increased risk, right? Which is, epidemiologically speaking, really, really almost negligible. It's just that there's lots of them, and so they can add up to something significant. When you look at the microbiome papers, again, just taking at face value the associations, and you actually look at the effect sizes, they're on the order of one of those single nucleotide polymorphisms in a GWAS, on the order of like 1.1, 1.2, that kind of effect size. So really, really, it's like, first of all, you should just treat them as noise based on the design. Secondly, whatever is there, could be caused by confounding that's probably, inconsistent across different studies. And third, it's not a big effect anyway. It's a tiny effect, right? So the idea but the problem is the headline doesn't say that, right? The title doesn't say that.
I was about to say.
The headline just says, microbiome differences between autism and controls, or the microbiome is associated with risk of autism, and then that just gets run with. That's what becomes the story, right? So that's
really well. Really well covered. I think what surprised me is that I can understand that the general population reads the headline and the effect size doesn't make it into either the article, or they just don't read it because it's 3 paragraphs down and people have lost their attention span 3 sentences in. But it sort of seemed like almost even the scientists that were building upon other studies also may have not fully taken into account the effect size. Again, that issue of the papers finding a difference, but not the same difference, and then the scientists taking that as replication is like, it's like, again, I can understand this on the level of like the standard, the layman, the individual who's reading headlines from BuzzFeed to get their science information. But like, the scientists shouldn't be making these mistakes. I mean, like, this needs to be.
Yeah, look, I'm not going to psychoanalyze the people, who've done the work, and to be charitable, I think they're well intentioned and trying to do good stuff, and but yeah, you're right. At some point, you can hide a lot of these inconsistencies if you just say that there's dysbiosis in these patterns. Any pattern, any difference is dysbiosis, and then you could find dysbiosis and I could find dysbiosis, and we could have exactly the opposite patterns of findings and both report dysbiosis in people with autism, right? So all right, there's some incentives in science, right, that are at play. And first of all, there's just kind of academic incentives. Well, no, let's there's a general motivation that people want to help, right? I mean, they're studying these conditions. They'd like to find something that explains them, that can eventually help people, even help treat people, right? So there's absolutely good motivations there from that front, right? At the same time, people want to have a successful career, right? So they want to get papers. And so they want to have their papers noticed. They want to get their papers into the best journals. They want to get them covered in the New York Times. And so there's those kinds of incentives as well that are at play. And scientists are human beings, and they are prone to those kinds of things. And they also want to keep their lab going, and they want to get the next grant. And so if I do a study and there's something there, it's a bit wishy-washy, but I can spin it a certain way that makes it look quite positive and sort of glosses over some inconsistencies, then that's my preliminary data for my next grant, and the next and the next, and even if I'm reviewing one of your papers and it's got some inconsistencies, but I can cite it in my grant, well, then, as a peer reviewer, I'm not impartial. Actually, if we're all in it together, if it's in all of our benefit to have this hypothesis seem to have good evidence, then I think that can lead to some peer review practices that aren't as rigorous as they might be.
Right.
And then the final thing is that in many cases, the researchers involved in these fields have some commercial interests. So their funding comes from big food companies, or from supplement startup kind of things, or probiotics and so on. They may have patents on particular things. They may be trying to develop them in biotech companies, and so on. So, those things are normally declared in the paper. There's a section about conflicts of interest, which is great. They're not always declared. I mean, there are a few cases in this literature where there were conflicts of interest that were not declared, but generally speaking, they are. And so
that's terrible when it's not.
Like it's very unethical,
very nefarious
when it isn't. But even when it is, there's this kind of attitude towards it in the literature. It's like, oh, well, if it's declared, then that's fine. It's all above board.
If someone's aware of their bias, they're clearly not biased anymore. By just being aware of it means you're not biased.
It's sort of,
that's how bias works.
Exactly. Yes, exactly, Evan. So, I think, for me, reading the paper, that becomes a consideration, right? If I find that the lead author or the senior author has a patent on the thing that they're proposing, is potentially involved in autism as a cause, or even as a therapeutic, and we'll get onto these ideas of what the therapeutics could be, I'm going to take that into consideration in weighing the evidence.
Right. I'd love to dive more into that, but maybe we should move along. I think it's a very interesting conversation on like what issues here are systemic in academia and what we could do about it. But let's maybe move to mouse model data, because this truly, I've always kind of been inherently skeptical of mouse model data, and maybe you can convince me here not to throw the baby out with the bathwater. But let's go into, like, what is a mouse model of autism trying to achieve? So
the reason that people have done experiments in mice is precisely because you can't infer causation from the observational studies alone, right? So people did some studies, they found some differences. They weren't consistent, but say I found some differences that I think are cool, right? Regardless of whether they were
Yeah.
replicated or not. I think they're interesting, right? And so I found, here's a few bacteria that are at higher frequency in people with autism than people without. So, a natural hypothesis would be to say maybe these bacteria, if I put them into a mouse, could cause autistic-like symptoms. I could test that, right? And it's kind of like the way people tested pathogenic bacteria like H. pylori that causes stomach ulcers, right? It was tested by, you take your supposedly pathogenic thing and you put it in a new host and see if it develops the condition, right? Now, that's sort of unstated, right? It's rarely actually made as explicit as that. What the conceptual thinking is, that's the foundation for these hypotheses. They're usually a little more loose. It's like, there's been some observational studies that have reported something. We think something about the microbiome is involved. Let's do something to the microbiome in a mouse and see what happens. That's sort of as explicit as it tends to get. Now, you might say, what should you expect to happen in a mouse that could tell you anything about autism? And so there's a whole field where people try to model some sort of human diseases or disorders in mice. And if it's like a liver disease or it's blindness or something sort of physiological like that, sometimes you can get a pretty direct correlate. Even, not always, because mouse physiology is different from human physiology, right? For psychiatric stuff, it becomes really difficult because a lot of our psychiatric symptoms manifest in very, very human specific ways, right? They affect our highest,
Right.
They affect language and mood and thought and, kind of very human-like behaviors, like, for autism, social interactions, which are so sophisticated in humans in terms of understanding people's body language and, these kinds of things. And as well as things like, repetitive interests and so on. So the symptoms of autism in particular are very human specific manifestations, right? And the question would be, okay, maybe that's true, but maybe they're manifestations of some neural difference that we could institute in a mouse where it would have a mousy version of those things, right? And then the question is like, how would you know that you had done that, right? And so there's a couple ways you could do that. One is to say, well, look, I'm going to do mouse tests of behavior that look like these human symptoms. So the most obvious one would be in social behavior, where you could say, okay, I'm going to see how sociable my mice are by asking, for example, how much do they investigate another mouse versus just a novel object, for example, right? And mice do differ in that sociability. And so people have taken that to be a kind of a model of an autistic-like behavior in the mouse. But it's a very superficial similarity. It's basically that you can use some of the same words to describe what's going on, and it's sort of a hope that there's a mechanistic basis that's shared. Now, how would you know that there is? There's a couple lines of evidence that you could take. One is you could take known risk factors for autism, things like high-risk genetic mutations, where we have dozens and dozens of them that we know about. You could cause those mutations in a mouse and then see if they develop this particular suite of behaviors. And there's social interactions, there's ultrasonic vocalizations that mice make that you can record with special microphones, and people take that as a kind of a proxy for communication in humans, which is, to me, a huge leap to go from squeaking to
these all seem like
language.
gigantic leaps.
Yeah, and then the other,
don't forget the marble
so
burying.
the marble
There's
burying is a favorite. This is one where if you put some marbles in the bedding of an animal's cage, they'll bury some of them. They like to dig around in the nest, and it's taken as this indication of sort of repetitive behavior. And that is like, well,
like what?
I know it's
is that not insane? Like, give me some sanity here, because when I read that, I was like, a genuine scientist cannot possibly compare a mouse digging in its bedding and a marble going down in the hay or straw to obsessive compulsive behavior and mannerisms in a human context. Right. I was like, that is so insanely stretched that it just It seems like almost I was being spoofed. Like
Yeah.
it sounded like a
I know,
joke.
because it's not a model of, say, stimming, where you get repetitive stereotypical behaviors, and it's also not a model of narrow interests, which is another sort of part of the phenotype of the symptoms of people with autism, is like really, really interested in train timetables or whatever it is. It doesn't matter, whatever it is,
Yeah.
right? Something. But
Yeah.
burying marbles doesn't seem to match even at the face validity level, right? Now, there are ways that you could get at it, though, right? You could say, okay, like I said, you make a bunch of the same mutations in mice and they show up with these same sets of phenotypes. It turns out that doesn't happen, right? So when you take the best etiological constructs that we have, the ones with the highest confidence, and you model them in mice, you just don't get a consistent panel of profile of behavior across these different tests, right? The other way you could do it is you could use a medication. So for something like schizophrenia, like for psychosis, you could say, look, this can be induced by drugs like amphetamine or ketamine or all kinds of other, psychotic drugs. I could give those drugs to an animal and see what behaviors it manifests. That's
Yes.
reasonable, right? Then you could say this is a mousy version of psychosis. And you can go one step further because you can treat psychosis, right? So you can give them
You can give it an
haloperidol
antipsychotic,
or clozapine or some antipsychotic and see if it reverses those symptoms.
Yes.
And then you would have good confidence that whatever your mousy phenotype is has some underlying neurobiological
It made a
basis.
prediction, right? It made a prediction.
Exactly. The problem for autism is there are no medications that treat the symptoms of autism. There are medications that treat, anxiety and other sorts of symptoms that go along with autism, but they're not the symptoms that autism gets defined by, right? So we don't have any access to any of that kind of evidence. So what we're left with is this sort of superficial similarity between these tests that's obviously highly arguable, right?
Yes.
So so with that as a background, right? Question: is there such a thing as an autistic-like mouse based on behavioral tests? Super, super questionable, right? The other question then is like, okay, well, what's the manipulation that these people are doing to test the microbiome? And what they're using is usually germ-free mice. And these are amazing, right? These are mice that have been bred in completely sterile conditions. So they have no bacteria in their guts at all. They have no microbiome. And then they're like a blank slate, right? So then you can add microbiome. You can treat them with probiotics. You can treat them with fecal transplants from other mice or from humans, and you can populate their guts, and then you can see now what's their behavior like, right? And so some of the really seminal papers that get cited thousands of times as evidence for a causal effect, we're taking this approach. The problem is, and it's really weird, is that some people in the field take these germ-free mice to already be kind of weird. Like some of them say, well, they already show autistic-like behaviors. Now I'm going to treat them with something and see if I can improve them. And then other people say I'm going to take them as normal. I'm going to administer to them, fecal microbiota from autism patients and see if I can make them autistic, right? So already there's this sort of lack of consensus in the field, even just about the experimental setup that they're using.
The second one definitely makes more sense. I mean,
yeah, it does. But the question is like, the germ-free mice, they do show some behavioral differences from regularly reared mice. It's just a question of what you make of that, right? What do you think your baseline is for these experiments? So there's a set of experiments that were done that either generated mice that they thought should be a model of autism, or took these germ-free mice and sort of tried to populate their guts. I think maybe let's just talk about the final one, because it's the most striking kind of experiment, right? So you take these germ-free mice and you give them a fecal transplant from either patients with autism or from healthy controls, so neurotypical controls. And then what the authors did was run them through a bunch of tests and do a bunch of stats. And they saw some differences in these things like marble burying and social interaction and so on. And there's a few things to be said. First of all, on the social interaction thing, what's interesting is they didn't see any difference in the test that's sort of taken in the field as the standard test for social interaction.
Should be the best one.
And what they did was then say, okay, well, let's try another test. Let's try a different kind of a social test, right? And there they found something. So it's this sort of a classic example of what's called p-hacking, right? Where you don't find a difference with one thing, and then there's enough degrees of freedom in how you're deciding what experiments to do that you can just keep going.
Yeah.
You can just keep doing. You can use this covariate or that one. You can vary these conditions until you find something. And clearly that's what happened in this case, right?
Yes.
But the other thing that happened with this particular paper is that they just did the stats wrong. So they had 5 patients with autism and 3 without. That was their samples that they took these stool samples from and did transplants with. And then they transplanted for each of those humans, they transplanted them into multiple mice. And then they treated those multiple mice as independent experiments as opposed to replicates of the same experiment. So they inflated their sample size for their stats way, way, way beyond what it should be, right? People pointed this out to them very quickly after the paper was published in Cell. And they, in fairness to them, supplied the raw data for other, researchers to look at. And when it's analyzed properly, basically all the results go away. All the major findings are just gone. There's no behavioral difference left. Maybe there's a little difference in marble burying that's barely significant, I think. Okay? So it's shocking that a paper that's so flawed, and it was obvious, like the minute it came out, as I remember, this was on when Twitter was in its heyday. This is in 2019. Many people just immediately looked at it and said, no, this can't be right. Those figures just don't look right at all. This wasn't done properly. And very quickly, within days, these flaws were highlighted. Right? The paper, of course, is still there in the literature. It hasn't been retracted or corrected. It's cited, I don't know, over 1,000 times, I think.
I think it was 1,500 from what I saw. I think if it's the Sharon and colleagues paper, I think.
And so, and then it's also just referred to in other studies, right? So you'll see other papers that say it's been shown that in mice, treatment with, an autistic microbiome, or that is a microbiome from people with autism, causes autistic-like behaviors in the mouse. And they just sort of take that as a given. It's become lore, right? And so what happens is, a student might read a paper like that. They see that in the introduction and then just say, okay, well, I presume these people know what they're talking about. I'm going to take that at face value. And then I'm going to say that, right? In my paper, I'm going to make that same citation claim. And then you get a citation of a citation of a citation. Like no one's gone back to read the paper and use their own judgment to evaluate what was going on there.
And if we're being like realistic, like how many more times does the finding just be taken from the introduction or the abstract rather than the student or even the scientist in a lot of cases does read the whole paper? It's far more common.
And in fairness, a lot of, I shouldn't say in fairness, but in actuality, a lot of the times there's a citation that does some work for somebody. They need a citation in a paper to bolster some line of argument or narrative that they're making. But anyway, ultimately from the mouse work, despite the way that it's been portrayed, there is no evidence, like zero evidence of any causal link between the microbiome and autism at all. It's just absolutely, to my mind, nothing there. There's no relevance to autism that's demonstrated. There's no clear findings. I mean, the sample sizes for some of these other papers, again, they were tiny and way lower than the recommended samples that have been put out by people in the field studying these things, who develop these behavioral tests, who say, look, you need multiple replications, 20 animals per group, per sex, per age, not 5 animals, right? So
5 animals. That's the one that I read. It's like I should at least have more fingers than there are subjects in your study.
Yeah, that's the frustrating
I was like,
bit of it, right?
But that's where it was kind of like, this seems almost too strange not to have some malicious incentives somewhere. Because I'm like, that seems too crazy. I mean, you don't have to speculate on this. You can allow me to more. But like, that's where I'm like, that seems too crazy to just be a decision that scientists made, that 5 samples is sufficient to publish. And then that goes on and gets 1,500 citations. Like, it's worth reinstating just how much body of evidence and body, or I should say body of science has been, came out from these types of results. 5 animals is completely
Yeah.
crazy. I remember reading that study and thinking, wow, that's so cool. I read colonization of mice with gut microbiota from human donors with autism spectrum disorder, but not from controls, is sufficient to promote behaviors in mice consistent with autism spectrum disorder. I was like, whoa, that's such an interesting finding.
Yeah.
Like, and then I wrote, and then the next sentence is, regrettably, this paper is beset by statistical problems. And I was like, God damn it.
Yeah.
The first paragraph was so interesting and so exciting. I was like, that's such an interesting finding. And then you realize it's 5 animals, and then there's just no statistical power to be heard of whatsoever.
Yeah, no, I think you've summed it up. To my mind, there's just nothing there. The mouse literature provides no evidence for this claim, despite the way it's portrayed and cited. Yeah.
Okay. So far the exploratory associations are almost certainly spurious, and then the behavioral findings that have been built on top of that just seem garbage, to be honest. You're so kind in your paper. I'm always wondering, do you have to be very cautious with the type of words that you use? Like, you can't just say it's garbage, even though really using five samples, to me, it's like that. There's actually no excuse for that. That's like, even pretending that science is kind of wild.
Well, yeah, I mean, I agree, but obviously we have to be polite and just factual. We tried to just not actually have commentary throughout about what our reaction is to these things happening, and just say, factually speaking, here's what these studies did, here's why they're flawed, here's why just conceptually they don't make sense. I mean, even these mouse studies, like we talked about the effect sizes, that were found in these observational studies. Well, what do you think is going to happen if you modulate something that has an effect size of 1.1 in a mouse? Why do you think that's going to do anything that you could see in the first place, right? So right from the get-go, the conceptual foundation is just not strong at all. And then, you can just say, look, they didn't, here's the sample size they should have. Here's what they did have. And, just leave it at that, and leave the meta kind of commentary.
For my podcast. Exactly. What? I'm curious, like, is there a sample size that you would have read and then been like, okay, well, that's a really cool result? Like, what sample size should you be aiming for in this kind of behavioral mice study when you have compounding variables, like how the mice is feeling on a given day, how much it personally likes to bury marbles, like how many mice is a good number of mice?
Well, so you've hit on some of these again, there's these confounds, and like mouse behavior is super, super complicated because mice are animals. They're not little robots, right? They do what they want. And so you're looking for patterns of behavior against this background of mice just being mice, right? And so people did some studies. So there's a woman, Jacqueline Crawley, who developed a lot of these tests. Really, really robust, rigorous scientist. And with a consortium of people, they took some of these mouse, genetic models where they have mice with exactly the same mutation, same strain in different labs, and then let them run through the same protocols. And what they found was actually very little consistency, right? So even the best people, the people who develop these tests, who go about it the most rigorous way, find that doing these behavioral tests across different labs, you got slightly different conditions that you don't even know could be confounds. I mean, a few years ago, that people discovered that the perfume that people were wearing into the test arena had an effect on mice.
Interesting.
So there's unknowns there. Yeah. So what's the right sample size? Much bigger than what people did. But I like to think of it in a different way. Like, you can have perfectly robust behavioral outcomes where there's a big difference that you see. And then, you might say, I mean, we've done this with genetic mutations in mice, where we're like, okay, their brain is messed up in a bunch of ways. Let's see what's different about their behavior. You run them through a bunch of tests, and you find one or two that show a difference. But you replicate that. You don't just take it you just don't take the positives as true facts, as findings, until you replicate it. So you can't just rely on bigger and bigger sample size. you actually have to just do a different design, replicate. And one of the things that's striking about the mouse work is, some of these studies were published 12 years ago or more. You would expect, if there was a really robust finding, that there would be a bunch of follow-up papers that explained that replicated it and then explained it and then got to the mechanism behind it, right? That's how science normally goes when something is robust. And instead, there's nothing, right? There's just a different design. If someone else comes along, does it in a different way, but the original finding, just kind of sits there. And then that just makes me very suspicious that it wasn't anything robust, because you can bet that people tried, right? You just don't hear don't hear about when they didn't go well. And that's the other kind of really pernicious problem, is publication bias, where you can do some experiments. If you find something, that's a story, that's a paper. If you don't find something, that's just a bummer, right? It's like,
put it in the
drawer. Yeah.
Never happened.
And so we don't know, right? That's the problem. We don't even know how many studies like that didn't replicate something, didn't find a result. That's the hidden that's the dark matter of the scientific literature that we don't get to hear about.
Right. Am I going too far by throwing all mouse models out? Because I remember I was speaking to a friend who's done psychedelics research, and they were doing the forced swim test for mice. And I remember him talking to me and calling me, and he was like, I'm just looking at these mice pretty much all day, and I'm trying to figure out if they're swimming. And sometimes they're like kind of slightly like moving a nail and kind of ever so slightly moving. And the idea is, they're trying to see, is the mouse swimming in the pool, and that's supposed to generalize again to some human
depression
characteristic of anxiety or depression or something, which again just seems completely insane to me, with depression being probably the most heterogeneous human contextual thing. And they're trying to see, and he even said to me, it's like, there's no way this would replicate. There's huge errors all over the place. Like, we're trying our best. We have this contract in from a Korean pharmaceutical company, and they basically told us that they want this result. So we're sort of, and like, they're not explicitly, but there's always a sense that that's the case. And then they're trying to interpret these results of like, is the mice swimming? Is it not? And the whole thing seems crazy.
Okay, so
like, it doesn't shock me that they would never replicate.
Well, first of all, just in terms of like mice as a model system for studying behavior generally, right? There's tons and tons of really great data trying to figure out, how the brain produces behavior, right? As models for psychiatric symptoms, that's a whole thing in itself. And some of them, I think, are more robust than others. The ones that are more robust are the ones that have this dual pharmacological evidence to go with them, where you can say, I can give some drug, it induces this symptom in humans, it induces this behavior in mice. I can treat it with a drug that I use to treat the symptoms in humans, and it rescues that behavior in mice. Then at least you have some confidence in the model, right? The experimental thing that you're doing. But then you still have to do it properly, right? And it still is the case, like your friend was saying, these are noisy data, right? So I think people are developing automated tools, behavior tracking tools that are much more robust than some grad student sitting by the edge of a pool with a clipboard, ticking off something, is going, did they rear? Was that on its hind legs? Or exactly. So it's getting more kind of reproducible by using those sorts of tools. I will say that in the pharma industry, there's decades and decades of research where people are using animals that have this kind of one of these sorts of a phenotype in one of those tests, and then throwing whole chemical libraries at them, trying to find new drugs to treat depression or new drugs to treat psychosis or whatever it is, without any success practically, right? There have been no new drugs with new mechanisms of action discovered in that way. So that's a real counter to what I was just saying about, having some trust in the validity of these things. The payoff would be you could use that as a model to test new drugs, and that just hasn't happened despite people putting loads and loads of effort, time, and money into it. So I think you're right to be skeptical of those kinds of things. in general, and we can be more skeptical for some kinds of things than others. I'm the most skeptical for the
Yeah.
autistic-like behaviors, as we discussed.
Yeah. There was a really good quote here that I have from the paper. Yeah. So clinical presentation of autism is highly heterogeneous, making it even harder to know what a mouse model of autism should look like, given that there is no such thing as a good human model of autism, just idiosyncratic exemplars of a fuzzy construct.
Yeah.
I thought idiosyncratic exemplars of a fuzzy construct was really well put, and sort of summarizes maybe everything we were just talking about there.
Yeah, and it highlights the fact that autism, like all psychiatric disorders, is diagnosed on the basis of patterns of behavior, right? There's no blood test, there's
Yeah.
no genetics marker, there's no brain scan that you can do, there's no biomarker of any kind where you could say, aha, now I have proof that this person has autism, right? In the way that you could do for, diabetes or something like that, right? You just don't have that because the nature of the condition is such that it just is a cluster of symptoms that seem to go together, and we put a label on it, right? And so the idea that, some people are advancing that they could use a microbiome profile to diagnose autism is so weird to me. Like, what is it you think you're doing? Are you going to run microbiome profiling on a bunch of people who don't have these symptoms and say, oh, look, sorry, your profile says you're autistic. I know you don't seem like it. Or are you going to take someone who has all of the symptoms and has a clinical diagnosis and say, well, no, sorry, you're not. You're really not autistic.
You're missing these 3 species of bacteria in your gut, so unfortunately you don't fit the criteria.
Exactly. It's just a category error. It's like, what are you? I just don't get what the idea is. But again, there's huge hype around those things. There's money that goes into those things, those diagnostic biomarkers, that's potentially big bucks. But it's like just a head scratcher. It's like, what do you even think you're working on here? It's just really weird.
2025 is the year that me and you are going to fix our sleep. Now, that does not mean that we need to go full Brian Johnson on it, but one thing we can do that's super easy is to buy the highest quality sleep mask on the market, and that is Manta. I've been blown away by how much better I sleep with one of these on. It's just a fact that people are not sleeping in completely dark environments, and studies show that that affects sleep quality, time to get to sleep, and increases the fragmentation of your sleep. I have tried other sleep masks. I first bought one on Amazon last year for like €5, but the problem is they're just really poor quality. They don't really block out any of the light, and you can't adjust them to your planet-sized head. Manta is fully adjustable, so it literally will fit all of the strangest and weirdest size heads out there, like mine. The Manta mask is literally completely blackout. I have studio lights coming at me from here, and I can't tell the difference. It is absolutely pitch black. This is the perfect environment that you need for sleep. Manta honestly feels like you're being cuddled by a cloud. I actively enjoy and look forward to putting these on each night. I just realized how boring that makes my life sound. I sleep with Manta every single night, and I've seen direct improvements in the quality, duration, and how rested I feel when I wake up. My Whoop data seems to agree. Manta have a bunch of different sleep masks on their website. I have tried the normal sleep mask as well as the sound mask. I like to listen to audiobooks at night, so for that reason, the sound mask was actually really, really good. For some reason, my girlfriend doesn't want to listen to how reality is a perception at 11 o'clock when we're trying to sleep, so this was a really good solution for that. It's also incredibly comfortable. But as I said, I think for most people, like 90% of people, the normal ordinary mask is going to fill their requirements. Super comfortable and completely adjustable. Literally is just a really high quality, well-made product. It's the best on the market. Manta are giving my viewers 10% off their first sleep mask. They ship internationally, and they also offer a 60-day free trial. That's 59 of the best nights of sleep you've ever had. I literally promise that. But if for any reason you aren't satisfied, you will get your money back, no questions asked. I really couldn't recommend more if you're looking to do something to improve your sleep. A sleep mask is the lowest hanging fruit. It's so simple and easy to apply, and I think it will benefit your life and overall health, and I could not recommend Manta more. Yes, Let's go to human trials. So human trials is the last sort of line of evidence. The most cited paper in this field is a microbiota transfer study. Can you explain kind of what happened there?
So this was a study that had 18 patients. I think if it's the one I'm thinking of, Kang et al. Probably. 18 patients where it was what's called an open label, uncontrolled single arm study, which means you just take a bunch of patients, you treat them with something and see what happens, right? Now, the problem with that is that, most clinical trials have a control arm where patients are treated with a placebo to control for the fact that the placebo effect is really real, right? If you treat people with anything, they may report that they're doing better, or in this case, their parents may report that they're doing better. So kind of a manifested wishful thinking, right? And it's just common, so common that's why we do control arms, right? And the other thing with that can happen is that there can be a genuine change in symptoms, but it's a sort of a regression to the mean where, they got enrolled in the trial because they were particularly, severely affected at the time. That's what motivated them going to the hospital and getting, enrolled in a trial. And then, there was just a natural fluctuation away from that, which tended to make them get better because they were at the extreme to begin with, right? So that regression to the mean is something that can just happen anyway. And then the other thing is that over time, and this particular study had a 2-year follow-up, you can just have maturation, right? So many children with autistic symptoms just get better over 2 years, right? Which is the span that they looked at. So
they learn their own coping mechanisms.
Exactly. That's just very common, right? So if you don't control for those things, then you're setting yourself up to find something that's not real, which is what this paper did find, is some improvement in some behavioral symptom domains. Again, not corrected for all of the tests that were done in this really, really small sample, where, in fairness to them, they said what should be done is placebo-controlled trials, right? But they also spun it obviously as a really big thing, and I don't forget how many citations that one has. Again, well over 1,000, I think. And again, it's cited as proof that manipulating the microbiome can lead to improvements in autistic symptoms. There were a couple other follow-up studies that were done. Independent studies that were done where, these were, if I didn't say it, these were fecal microbiome transplants from healthy individuals, right? So the concept is, well, something is amiss. There's some dysbiosis in the microbiome of the patients with autism that is causing some of their symptoms. And if I transplant in a healthy microbiome and it engrafts there, and then it can change that microbiome landscape and ameliorate the symptoms, right? That's the concept, and that's the interpretation. So a couple other people have tried.
Maybe worth mentioning, just because this was a really complex treatment. Like, it involved 2 weeks of antibiotics before the implantation. So it's quite a complex treatment. I guess the idea of the antibiotics is to almost level the playing field
to wipe out the microbiome that's there and then replace it with something else. So the incoming ones don't have to compete, right? And it was replacement with this, I forget the name that they use. It's a sort of a TM.
Standardized human gut microbiota, which I did not know was a thing whatsoever.
Right, that's sort of a proprietary thing, I think. Anyway, whether it is or not, it's not taken from an individual person. It's some sort of smoothie made out of patent
pending blend.
It's a special, a unique blend of So a couple other studies were done with that same design, where it's an open label. I mean, everybody knows what's happening. It's not blinded, and it's a single arm, so no control arm. And again, they reported some improvements, not exactly in the same thing, so a little inconsistency. But then a randomized control trial was done with much, larger samples, I think 50 something per group, and they found no improvement. Right? And so this pattern just recurs because there's another kind of manipulation that people do, which is not a fecal microbiome transplant, but just treatment with probiotics. So probiotic just means a bacterium that is taken to be beneficial in some way. There's a really vague term.
It's pro. It says pro right in the word.
Exactly.
There. It's got to be good.
Exactly. So probiotics
right there in the word.
Probiotics go. That's the That's the good thing. I don't know. But I mean, it's funny that's a term. It's not a scientific term, really. It's a very judgment-laden kind of a term. But anyway, people have used various things that they took to be probiotics, either single species or combinations of them, and treated people with autism with them, and looked at the results, right? And the exact same pattern emerges. Small trials, open label, single arm, often report something. And then when it's done properly with controlled trials and control arm, blinded, then they see nothing, pretty much.
Yeah.
And so there's loads and loads of these trials have been done, meta-analyses that have been done, which just consistently find no evidence for probiotic treatment in ameliorating the symptoms of autism, or for fecal microbiome transplant treatments either. So the human trial data, I mean, the overall conclusion from them is that when the experiments are done properly, you don't find any effect.
Yes. Surely if you do a study that doesn't control for placebo, which is just such a well-documented phenomenon now in basically everything that expectation drives result, that paper should only be used as a signal to drive then a placebo-controlled trial, and then the result of that experiment needs to be appended to the original study in some way. It needs to be edited after the fact to say, like, you can't just say a placebo-controlled trial needs to be done, but these results are promising. When the study actually is done, surely it needs to be put into that conclusion in some way.
I would love to see,
because people just read that. People just read the initial, like, oh, that's really cool. Right. I wonder, did anyone do the placebo-controlled trial? But I'm not going to look it up because this, confirms my bias.
Right. Yeah. I would love to see that practice, right? Where, but there is this thing in medicine where people do these exploratory trials with small numbers of patients with very suggestive ideas, right, in mind. So these hypotheses that don't have a lot of support behind them, but maybe you can do this small scale trial and get some support for a larger study. I think that's the way that people think of it, right? And it's fair enough, but it would be really nice to see that retrospective linkage in the literature somehow, where that people after the studies that come out that do the control trials stop paying any attention to the other ones, like stop citing them.
Exactly.
They were clearly spurious. So now we know.
That's my point. Exactly.
Yeah. So the fact that these studies keep getting cited, it's not like they slow down at all, right? I mean, this is a just huge citation where people take these 3 lines of evidence. It's not just that they take each of them to be solid, it's that they take the 3 of them to be reinforcing each other, right? It's like, how could it be? Yes. But look, you're ridiculous. There's observational studies and these things in mice and these trials in humans. And sure, maybe each of them is a little dodgy, but it couldn't be the case that that's just a coincidence, right? But yeah, it can be, because there's just loads of noise in all of those areas. And there's things, like I said, there's studies where you don't have to rationalize them or figure out or interpret them, right? You can just ignore them, right? Yeah. Once once studies are done with thousands and thousands of people and they don't find something, then you don't have to go back to the ones that were done with 10 people and say, well, okay, but how do you make sense of that, right? You can just stop talking about it.
Right.
Yeah. So that there's this sort of pseudo triangulation that happens between these lines of evidence that makes it look like they support each other when actually they're not even commensurate, right? They're not even comparable conceptually with each other. Like, we talked about the observational studies and the mouse studies, and the gap, the disconnect when you think of the effect size. It's like, what are you even doing there? Like, what do you think is happening?
Yes. Yeah, it's super interesting. And I think as a paper gets more citations, it's sort of a self-fulfilling prophecy. It's almost social proof, and it gets more citations again and again. It's like reviews on Google Maps. It's like it's taken as some signal that this restaurant or bar is good if the paper has lots of citations. And then you never see the non-replicated evidence or the actual placebo-controlled trial that that didn't have a significant finding.
Yeah. That's a well-known effect, a documented effect in the scientific literature, is that citations. Papers get a life of their own based on people citing the citation, basically.
Giant news. We created a completely free ebook titled 26 Neuroscience Books for 2026. I've read over 100 neuroscience books in my life, and with Christmas just around the corner, I thought I would help people find the perfect book for them or to buy for someone in their life. It includes really nice science-based illustrations, an accessibility guide, and personally written segments for each book and the impression they left on me. I also asked 100 top scientists like Dr. Mike Levin, Anil Seth, Nick Lane, and many more what their favorite science book is, and you can find all of those included at the end. If you want to support the channel, you can also purchase a beautiful physical paperback copy, also a perfect stocking filler. We put a lot of effort into our first Giant Shoulder product, and we cannot wait to hear your thoughts. Check it out in the link below. Again, it's completely free.
Right. What would be the problem with enforcing something like that, where if a study didn't control for placebo and it kind of gave this, it was exploratory, it said now placebo-controlled studies need to be done, and there is one that's done and there's no significant finding, like what would be the issue with enforcing some sort of appending onto the paper? Now, I could understand scientists won't like, but if they're being genuine scientists, they sort of want their finding, and maybe they don't want their finding to be not replicated, but it's at least good science. Yes. And then we wouldn't run into this issue of everyone citing the exploratory study that's underpowered and then nobody seeing all of the really good larger studies that aren't. It's such a clear issue that surely has some solution.
It's really tricky.
That we could work
the
towards.
Problem is that, like, no one is curating the literature, right? No one's in charge of this. This is a big self-organizing process system, right? And so the idea of enforcing something is like, wait, who do you think is the enforcer? There is nobody who does that job. This is a collective enterprise where what we try and do is hold each other to account and develop the most robust consensus that we can, at least when a field is functioning healthily, right? That's a healthy, normal way the scientific field functions. But at the same time, there's counter incentives that weigh against that. And we talked about some of them earlier in terms of people's careers and even potential commercial interests, for these probiotic studies and things like that. That's, people do have patents on those things, and they do have biotech companies developing those. And they do stand to make money out of those things. So it's in their interest to have those results sit there and not be directly contested in that way. So I think, again, what it means is that as a consumer of this literature, right, as a reader of that literature, for scientists, that they should be a kind of a buyer beware attitude. The problem is that's like, you and I can do that, right? And I mean, but it takes work. You have to dig into these things, right? It's not trivial to dig in and see where the problems lie. So even for scientists, it's difficult. For journalists, it's almost impossible. They just have to take the word of the people doing it, and maybe they'll, call somebody else for a quote that ends up like 6, 7, 8 paragraphs down in a study with a headline, fecal microbiome transplant treats autism, and somewhere down in the headline it says, Kevin Mitchell from Trinity College Dublin, who wasn't involved in the research, cast doubt on this thing, and there's a quote from me, right, or somebody else. That doesn't do a lot of good, because the general public then can't, they really can't evaluate the scientific details, and they have to take the journalist's word for it. So it's an issue, right? I mean, I think, particularly in this kind of case, when things are at stake here, like, this matters. It's not just some scientific theory that people are having a fight about that doesn't mean anything. It's super important, right? I mean, and people with autism and their families, in many cases are quite vulnerable. They're vulnerable to being taken advantage of. And there's so many quack cures out there that people are selling to people with autism and their families, that I find it really irresponsible to hype 100%. Something where the evidence for it is just not there yet.
100%. I went searching for probiotics for autism, because I was just curious, was anyone branding it in that way? And I couldn't find a specific product that was overtly advertising it like that, but I did find a bunch of websites that were like, what are the best probiotics for your autistic child? Like literally that exact branding. Loads of help sites with Amazon links. And then you went on the Amazon links and they were just probiotics for children. But like, you got a sense that the way that they were marketing and advertising them was targeted towards that group. Now, there's probably laws against them overtly saying probiotics for autistic children, but they pretty much did as much as they possibly could without overtly stating that. And again, who knows if they likely did create the web page that sent them to the Amazon link. You know, all of the different web pages that aren't bound by the same laws, like, you know, that really are targeting parents that are worried about their children and worried about the adverse effects that their children are suffering, and then they're being sold stuff. It's just it's gross on so many levels, which is why I think this paper is so important that you did. How long did it take? Like,
it took a while. We really did a deep dive, and you know, it's hard sometimes with some of these papers to even figure out what they did. You know, sometimes it's just really not clear. Well, like why did they do this analysis? Where are those data? What's you know, there's lots of stuff missing. It's not so. You just have to go into it in detail to try and figure out and make these comparisons. So, you know, thankfully my co-authors, Darren Daly from University College Cork and Dorothy Bishop, who's from Oxford, you know, they're both really expert in reproducibility and statistical analyses and clinical trials, clinical trial design, and so on. So we had a team, you know, of people who are, you know, has some expertise in the reproducibility area, and who are, you know, otherwise didn't have a dog in the race, you know.
It's enormous amount of respect for the amount of groundwork that needed to be covered to go through all of those studies. I don't know how many studies are that you go through, you know, methodology by methodology in this paper. It's very rigorous. It makes an incredibly convincing case, but like, more power to you. I couldn't, like, that just having to look through a literature of garbage and then just trying to, like, it just seems like there's so many, it's so important, but like, God, I would rather be doing literally anything else, like walking on nails.
Well, that's partly, you know, I mean, this is why it took, I mean, I've been wanting to do this for a long time, but it took a while to get the motivation up for it, to reach the degree of exasperation that made it, you know, feel necessary, and as well as hopefully worthwhile.
Do you have any other fields in your crosshairs? Are there anything else?
Well, you know, I don't go around spending my time just looking at stuff and trying to knock it down. You know, I don't like to read bad research. I like to read the cool, interesting stuff. That's how I like to spend my time. Having said that, there are some other
here's my kill
there
list.
are some other fields. So, I mean, one of the fields that has really similar issues with it is transgenerational epigenetics. So the idea that something that happens in one generation can have knock-on effects in like 2 generations below, right? So there's
super interesting.
So I've written, actually, you know, I did go into this in detail over several years, and I've written a bunch of blog posts about it that really look in detail at, you know, these human studies, mouse studies, and so on. They have all the same problems that we've just been talking about. Same statistical flaws, same sort of conceptual lack of depth, or lack of a really firm footing for even like what people take themselves to be doing, and also the same phenomenon in terms of public interest, right? It's this massive public interest in this idea that you can get these transgenerational effects, just like there is with the microbiome. And there's something behind that, I think, which is that these are radical ideas. They're sexy, you know. That's just super interesting. It's like your psychology is not just caused by the way your brain is working. It's in your gut. That's cool. Or your your phenotype is not just due to your genetics and your local environment. It's something that happened to your grandparents. They have this memory that gets passed down of being in the Holocaust, or the the Dutch
Right.
hunger winter. That's cool. That's a cool story, right? And so there's
The Irish famine is an
Oh, yeah.
example that's often brought up of this. Do the people of Ireland suffer any trauma from the famine, which
Well,
is
they suffer
300 years ago?
They suffer trauma. It's not that long ago. They suffer
Yeah.
trauma psychologically and sociologically,
Yeah.
right? And so
Yes.
there's loads of evidence that you get these sociocultural effects from historical events that get passed down through generations and manifest in important ways. You just don't need to say that that's mediated by some molecular chromatin epigenetic structure. I think people reach for this scientific sounding explanation as if to validate the idea that you could have these
Yes.
long-lasting sociocultural transgenerational effects. You don't need that. Of course you can have those
Yes.
kinds of effects. It's sort of it almost trivializes the phenomenon that you're after, I think. But anyway,
Really interesting. I wasn't familiar with that. I didn't know that. I thought that transgenerational epigenetic effects were pretty solid. So that exposes my ignorance.
Well, that's the thing. That's the narrative that's out there, and they're solid in plants and in nematodes, and just not solid at all in mammals, especially not in humans. But yeah, it's an example. It's just like the microbiome one in that way, is that you just get the impression from reading lots of stuff that there's this big solid body of evidence. There's a phenomenon that's well known, and then what we're doing now is trying to figure out the details. But you don't think to question the whole thing, right? The whole basis for the whole claim. Which is where we end up here.
Right. Kevin, this has been so much fun. What are your main research efforts at the moment? What are your cool science, not kill list type stuff? What cool science are you working on at the moment?
Yeah, let's see. There's a few things. There's one a neuroscience psychology thing where we're looking at the speed of sight. So different species have a different visual frame rate. It's kind of a tempo of visual resolution. And you can see that because you can look at different species can see a light flickering, for example. So the fluorescent lights above me here are flickering, but I don't see that because they flicker too fast for my resolution, right? But other species could see that, right? That's why insects like to fly around them and stuff. Okay, so really fast-moving species like peregrine falcons and things like that have super, super high visual frame rate because they're moving fast and they're tracking fast-moving prey. Turns out, so humans have an average for their visual frame rate, but there's huge variation. So what we found with Clinton Harlem, who was a graduate student, and some colleagues here, found that different people vary in this trait quite a lot. So the average is about 45 hertz. So that's when, if you have the way we measure this is like you turn a dial on a light that's flickering, and you change how fast it's flickering until you can no longer see it. And that's your sort of threshold, right? And the average is about 45 hertz in humans, but the range is like 30 to 60 hertz. Like, so literally some people are getting sampling the visual world at twice the rate of other people. And so what we hypothesize is that might, it's not going to make a difference in most of our daily lives walking around the place. But it might make a difference if you're trying to hit 100-mile-an-hour fastball or a cricket ball, or you're trying to return Novak Djokovic's serve, or something like that.
I was going to use the tennis example as a fan. That's so interesting.
Yeah,
that's really
cool. So our
cool.
hypothesis is that athletes who are performing in sports where they have to do high-speed tracking of visual objects could either be selected for a high level of that is, you might need to see the world fast in order to progress to become a professional tennis player or baseball player, or it might be trainable. So so that's one of the things I'm working on. And there's a few other bits and pieces.
I can probably serve a near to 100-mile-an-hour serve if you need any test participants. I've played tennis my whole life. I've been a coach.
Can you hit?
If you need me around Trinity to nail a few cannon serves, I'd love to contribute.
That's outstanding. Can you hit the 100-mile-an-hour serve?
With about 50% accuracy, maybe.
That's pretty good. That's pretty good. All right, we might get you over and test you and see how you do.
That'd be so cool. That'd be so cool. Kevin, it's always an immense pleasure. I couldn't be more appreciative again. Thank you so much. I think the paper is really, really interesting, really important. I am again, so much respect for just slogging through literature for hundreds of hours to get it done, but incredibly important. You're protecting, I think, a vulnerable population. So
well, thanks.
Couldn't be more appreciative, and always a fun to
I
chat.
appreciate it a lot, Evan, and the chance to get the word out on this, because it's not, oftentimes a sort of a paper with no findings or conclusions or negative conclusions. They don't often get the attention that the initial studies do. So so I appreciate the chance to talk about it.
Of course, the channel will do its best over time. It should accumulate some views.
Great.
Thank you so much,
All right,
Kevin.
thanks a lot, Evan.
But there's no evidence that in fact, I think we've kind of given up on the idea of evolution. The theory of evolution as articulated by Darwin is kind of not true. Right?
In what sense?
The degree of confidence with which he outs this ignorant bullshit really requires some mental discipline to reject all of the evidence. It's bothering me a lot lately because there are very strong claims being put out. There's a Netflix special on it, making claims, for example, that the gut microbiome involved in things like autism, depression, anxiety, PTSD, whatever. I mean, anything you can think of, there must be something going on. But it's all, as far as I can tell, and I mean, I've looked in detail, bullshit. I haven't seen any convincing thing whatsoever. We could search depression and microbiome, and we could pull up the top cited 10 papers.
Yeah.
I guarantee you all of them would have those kind of methodological problems with